on in Huntington's disease, for reasons that we don't fully understand. But what we know is that the cortex, the wrinkly bit, produces these things called neurotrophic factors, which are like fertilizer. So they produce it, but they pour it onto the striatum, and that nourishes the striatum and helps to keep it healthy and keep it alive. And in HD, the amount of neurotrophic factors is significantly reduced. So this is a very sensible target for Huntington's disease treatment. And the way that these neurotrophic factors work is that they land on these things that are like a baseball glove. Where's Jimmy gone? You missed my American reference. So the BDNF comes in and it lands on these receptors, and that then sets up these kind of health signals in the cells. That's difficult to do because BDNF is a huge molecule. So again, we'd have to pour it into the brain down tubes, and it wouldn't get very far, and it might cause immune reactions and things. So instead, what we're working on, what CHDI is working on, is these drugs that will mimic the effect of the BDNF or the neurotrophins, but will be able to be taken as a pill. And that's a process that's going on, and they've come up with some challenges. We're basically in the early days of understanding how these receptors work, but there are already a few candidates that are being tested. And again, this is earlier in the pipeline, but an extremely well validated target that's being worked on. Now, inflammation is the subject of my fourth of the top 5. So I need to introduce you to the idea that you've heard of the immune system, right? And there's the white cells that whiz around our bloodstream and they fight off invading organisms and things. The brain has its own immune system. So the orange stuff in the background here is the neurons that kind of run like fibers. And then these microglia are the brain's immune cells, and they kind of wander around the brain, clearing up damage. They do that through a combination of things, but 2 of the molecules they produce are called quinolinic acid, which is bad or dangerous, quina, or quinurenic acid, which is kind of good. So quin, when it's released from microglia, harms neurons. Quinurenic acid nourishes them, supports them. And the balance of quin and quina, normally quina wins out. In the HD brain, there's too much quin, and quin wins. And the enzyme that decides whether the microglia make quin or quina is called KMO, and it's overactive in HD. So what we need to do is inhibit that enzyme. And this came from working in yeast cells, believe it or not. They looked at which genes in yeast were most important for keeping them alive when they also had the HD gene. And it turned out that the KMO gene, which no one had ever heard of really, was important. And then independently, our group in London looked at the blood of HD patients and found that there's differences in the way the immune system works in the whole body in HD. And we then looked in the brain and found that those differences are also present in the brain. And that gives us very strong reason to suspect that KMO inhibition is a promising approach. And indeed, it's now 2 years since the first trial of a KMO inhibiting drug took place in mice. Now that drug doesn't get into the brain, and it's not a great drug, but it worked pretty well in the mice. But in the meantime, CHDI has been working very hard on these KMO inhibitors and has just announced this year its top candidate, CHDI 246. It's done all the things it needs to in the animal models, and most importantly, perhaps what they announced earlier this year was that when you look at the spinal fluid of animals that have been given this drug, the sorts of changes that you would need to see are observed in the spinal fluid. And what that gives us is a direct connection to humans, because we can look in the spinal fluid of humans, and that will hopefully give us an early way of figuring out whether these drugs are going to work in human patients or not. And those trials are now being planned. So that's pretty cool. The final one is a bit of a mystery, and I include it not necessarily because this is going to yield treatments sooner in the next year or so, but because it's a really good example of how far we've come as a community. So we talked about DNA, RNA, protein. What can happen, though, is that some proteins can get into the nucleus and change the balance of which genes are switched on and which genes are not switched on. And our DNA is actually protected by being wrapped around molecules called histones. And that can help to protect the DNA against interference. And the enzymes that control whether or not the DNA is protected are called HDAC enzymes, which stands for histone deacetylase enzymes. But don't worry about that. These are enzymes which decide whether our DNA is protected or not. switching. So there's these drugs called histone deacetylase, or HDAC inhibitors, and based on genetic work, one of the top HD researchers, Jill Bates, identified HDAC4 as the most promising of all of the HDAC enzymes as a means of slowing down the progression of Huntington's disease in animals, in mice. So HDAC4 is the one that CHDI is now focusing its efforts on. What's interesting is that the more work they do on HDAC4, the more mysterious it seems to be as a drug target. So they've now designed a drug that binds to HDAC4 in the right place, or rather the place where the enzyme's activity is. And the drug did what it was supposed to do, but the animals didn't get better, which is a real puzzler. But what it means, I think, is that there's something else that the enzyme's doing. It's got a second function, which we need to target. So it's not quite back to the drawing board. This is a good example of where you don't get the result you want, but that helps us to understand the nature of the problem. And we now can step to the side and find a different way around that particular obstacle. That project led to this slide being presented in Venice at the CHDI drug meeting earlier in the year. And this was really inspiring to me. I have no idea what it means. But it's the first time I'd ever seen a slide like this, and look at it. It says things like warhead, aromatic or aliphatic linkers, lower pocket, ADME handle, surface pocket. No idea what it means. But it's the language of drug developers. And if you go back 10 or 20 years and show a slide like this at a Huntington's disease meeting, people will go, what's the matter with you? Are you insane? Have you gone crazy? We don't do that. The drug industry does that. We can't. We're academics. We're trying to figure out what this gene does. This is what our meetings are like now. This is the problem. This is the drug, and these are the problems we've come up against designing a molecule to engage with that drug. So, as well as all of the research that's always been going on in the past 5 years, we have top drug hunters, drug designers working on Huntington's disease who understand this kind of thing, and it makes them quite excited, actually. So that's pretty cool. The bottom line is that there's lots of exciting stuff in the pipeline. Now, that was Act 1 and Act 2. Act 3, I reckon I've got about half an hour. I won't need half an hour. I'm going to sit down for 13 minutes, and I want to show you a video. But in case that upsets you, the idea of not listening to my voice for the next 13 minutes, it's a video of me. But it's not. That's not me. That's the other person in the video. This is the coolest thing that I heard this year. This isn't going to lead to a drug treatment in the next 5 years, but this. Anyway, I don't want to spoil the surprise. And some of you may have seen it already on HD Buzz. But to me, this says so much about the spirit of the HD community and the way that we engage with problems. So it's me interviewing a Californian researcher called Gwen Owens. That's 13 minutes.